Welcome to Lecture 2.3. Lecture 2.3 today is going to be on chemical reactions and enzymes. So as we go through today's lecture, keep thinking about atoms and molecules. We talked about reactions in that chapter when we looked at dehydration. What we're going to look at, or what we looked at next, was macromolecules and how molecules work together. One of the molecules we looked at was proteins. And proteins do work in the body. They are the molecules that actually do work. One of those molecules, to be more specific, is an enzyme. And what we're going to be looking at today are how enzymes function. You guys will demonstrate this in class, and then we will talk about it before we take our test. So today, enzymes. We're going to start out with, what is a chemical reaction? A chemical reaction is a change in the chemical bonds of atoms or compounds. So we're talking about if A is bonded to B, okay, and C is bonded to D, and we're just using these as like, example molecules. The chemical reaction would change those bonds into these bonds. Okay, what we are doing is breaking one set of chemical bonds and building more chemical bonds. The chemical reaction will do something like this. Now, it can either be just breaking, or it can be just building, or it can be both, which is this example. So what we're going to be looking at are some different examples today, but this is kind of the gist of it. Change in chemical bonds. The different parts of a reaction are as follows here. The parts you start with, the molecules at the beginning, are called the reactants. Those elements enter the chemical reaction. The products are the molecules that come out of the chemical reaction. So if you think of it as kind of like a box, the reactants will go into the chemical reaction box, and out will come the products. The arrow in any chemical reaction is always pointing towards the products. So when we look at chemical equations, you can see which one's the product by which way the arrow is pointing. Here are some examples. Here's one very famous example. What we call this here is photosynthesis. Okay. Photosynthesis occurs in plants. We'll talk about this more throughout the year. But the reactants for photosynthesis are very simple. Carbon dioxide and water. And actually, there is one, funny enough, missing. And that one is sunlight. So those three things go into a plant. Here's our plant right here, this arrow. And then out comes the products. And the products in this case are sugar and oxygen. So plants take in the CO2 as a reactant, and they pump out sugar, which is the product. And again, here the arrow is pointing towards the product. You can tell which one is which. The reactants are on our left, products are on our right. Here's a kind of pictorial depiction of what's going on too. Now, if we were going to look at just enzymes in particular, like I said in the beginning, enzymes are proteins, one of the four macromolecules. So we've already kind of talked about in brief what proteins do. Enzymes in particular will speed up a reaction. Okay, so when they speed up a reaction, what they're basically doing is taking the amount of energy needed to get it going, think of like starting your car, and lowering that amount. They're making it easier to get the reaction going. Okay, so what an enzyme will do is lower what's called the activation energy, and the activation energy is the energy needed to start. Okay, we'll get an example here of that in a second. Enzymes are also reusable. Make sure you remember that for later. Enzymes are reusable. So they do not go away after the reaction's over. They just sit there and wait to be used again. Like I mentioned earlier, activation energy is the amount of energy it takes to get a reaction going. So let's do a little hypothetical example here. Here is a big hill. All right? You want to go sledding. So, in order to go sledding, you are standing at the bottom of the hill, and in order to get up there, you've got to input some energy. That's called the activation energy, okay? So you walk and march your way up to the hill with sled in hand, sled. And now that you're up there, you can slide all the way back down and have fun and go through the chemical reaction, okay? So the activation energy needed to get up that hill is quite a lot of energy. Now, what an enzyme will do is a bit different. Let's erase this. Let's say you're at the bottom of the hill, but you still want to get to the top of the hill. You just want to use less energy, right? Well, guess what? If you hop a ride on a wagon or another sled, so I'm going to draw another sled here, and let's say you ask your dad to pull you. If your dad pulls you up the hill, and now you're at the top of the hill and you can go sledding, you've expended a lot less energy, okay? Using an enzyme is like asking your parents to take you up the sledding hill instead of walking yourself. You still get up there, you can still do the reaction or have fun, but it takes a lot less energy. Okay, so that's the idea behind activation energy. How much energy is needed to start a reaction? In here, you can see a very technical drawing of what's going on in a chemical reaction, but we've got our reactants right here, we've got our products, and the amount of energy needed to get going is this red line, okay? This is with no enzyme, so, okay? If an enzyme is used, the amount of energy needed to get going falls, okay? So with an enzyme, it's a lot easier to do the reaction because less energy is required to get up the hill and then make the products. So that's now, when an enzyme goes through a reaction, it reacts with very particular things, okay? So let's an enzyme is the protein, and the substrate is what is being used, okay? So the enzyme has to work with something. What it's going to use in a typical reaction is called a substrate. Back to the plant example, CO2 was our substrate. Plants took in the CO2 and then changed that into sugar. In this example, we're just using a square and a circle. That's still okay. Our enzyme is going to take that in, and then through a hydrolysis reaction, which we know breaks, the enzyme is going to bond with the substrate and break those reactants into separate products, okay? The special location, the last thing you got to remember about enzymes. Enzymes bind with substrates, and then through a typical reaction, which you know, you've learned about, hydrolysis or dehydration, you either make new products or you break new products apart. The way these enzymes fit together forms almost like a lock. Think about it that way. You have a lock. Lock is going to be the enzyme in this example. There's only one key that you can use to get into your house. Okay? The key is the substrate. So when a substrate comes by an enzyme, if the substrate has a different shape than the enzyme does, they won't fit together, and thus they won't work, okay? Enzymes fit together like a lock and a key. So there's one particular substrate for one particular enzyme. And if they don't match, then no reaction is going to happen, okay? Again, each substrate has its own enzyme. So we're going to practice this in lab at least twice, actually. But it's key to... All right, finally, your enzymes are affected by two different things, okay? Enzymes need a pretty consistent temperature. In fact, 98.6 degrees Fahrenheit. That's your temperature. So enzymes in your body will work very well at this temperature. If you raise the temperature or lower the temperature, the enzymes don't work anymore. Enzymes fall apart. What do we call that? We actually call that a special word called denaturing. Okay. The other thing that can affect an enzyme's function is pH. Okay. The pH in your mouth is a lot different than the pH in your stomach. Your stomach is full of acid. Okay. So when enzymes fall into your stomach, they fall apart very quickly because the stomach acid breaks them up. So we'll add this in here. Stomach acid breaks enzymes. Okay. So pH can affect it, temperature can affect enzymes, but these things have an effect on whether an enzyme will do its job or not. All right, so to finish this off, I guess, and sum it up, you use enzymes to release energy and use energy. When you break the bonds of sugar, you release energy. You can use that energy to do things. Some of the energy you make is released as heat. That's why you're 98.6 degrees. But some of it is used to make your proteins work, and that's why you can move your arms and listen to me talk to you right now and possibly even write down some notes. Okay, so energy comes from these reactions. Okay, what does this mean for you? Well, again, life requires reactions. We've already talked about that. Remember, one of the characteristics of life was having a metabolism, being able to use energy. How do you do that? Well, you first have to eat food. Okay? After the food goes into you, you're able to do things. Okay? Whether that is, go skateboarding, or write notes, like you're possibly doing right now, hopefully, or even, talk with your parents. All of those things require energy. So enzymes help you do that, and we can't really function without them. All right, so we're going to talk about this in class a little bit, do a few labs, but if you have any questions, come and see me, and I will see you next time. Thanks for listening.